Okay, good morning, everyone, or good afternoon, if it is where you are. Welcome to today's webinar for the Human Islet Research Network, or HIRN. And today, our topic is going to be stem cell-based platforms to model human autoimmune diabetes. I'm Joyce Nyland. I'm at City of Hope in the Beckman Research Institute and a co-PI on the HIRN Coordinating Center, or HIREC. And our presenters today are going to be Audrey, who's at the University of California, San Francisco. And we also have Karla Leavens, excuse me, who's at the Children's Hospital of Philadelphia. And once I get going, it doesn't want to go down.
Till April.
And if you could all mute until you want to ask questions towards the end. So just to give you a little bit of background, some of you know this already, but the Human Islet Research Network began in 2014, and it consists of now 5 consortia. The newest one is up at the top there, HPAC. That's the Human Pancreas Analysis Consortium. And then we have the Consortium on Beta Cell Death and Survival, Consortium on Modeling Autoimmune Interactions, the Consortium on Human Islet Biomimetics, a consortium on targeting and regeneration. And then coordinating all of this is the Human Islet Research Enhancement Center, or HIREC, and I'm the co-PI along with John Cadiz. There are quite a few people involved in the consortium now. It's freezing again, like this. Why it does this? Over 200 participants, over 9 different countries. And I think I'm frozen again. Okay, there we are. And it involves 85 different institutions. If you would like to read more about it, we recently published a series in the molecular metabolism. This is the introductory paper, and there's a number of others. Dr. Parent here is one of the co-authors. And if you would like to learn more, you can always go to our website. And the executive summary for year 5 and 6 has been posted out there now. Also, if you go to the resources tab, you'll find links to many useful resources like the HPAC-PancDB database, IIDP for islets, NPOD for tissue, Pancreatlas for images. And we also have a resource browser that gives you many different resources for diabetes research. And the webinar series, such as this webinar, will be posted out there. And particularly the resource browser is a rich library now of resources for you in conducting your research. We just had release 4. Here's the website up at the top, resourcebrowser.hearnnetwork.org. And we're into our new release, and we now have close to 2,500 different resources cataloged here, searchable antibody cells, data sets, tissues, all sorts of resources that I hope you will avail yourself of. And in particular, there are many human pluripotent stem cell lines posted here, which is going to be the topic of today's seminar. We also have special collections out there, such as tetramers, tested antibodies, humanized mice that have been reviewed. And after each webinar, they're posted on the website as well. There's a YouTube, and today's webinar will be there soon if you want to review it or tell your colleagues about it, or if you happen to miss it and want to go back to it. So today's webinars involve the following. Dr. Parent and Dr. Leavens are going to talk about the advantages of stem cell-based platforms to model human T1D, the current state of differentiation of T1D relevant cell types, examples of disease modeling applications using stem cell derivatives, and development of multi-tissue platforms combining islets and T cells. Just a few little reminders. The presentation is being recorded, and as I mentioned, will be posted at the end on YouTube. If you could please mute your audio and turn off your cameras during the presentation, and then we'll have a Q&A at the end where you can please raise your hand and unmute to ask questions. So with that, I'll turn it over to Dr. Parent. Let me stop sharing.
Okay, thank you, Joyce. Let me take a second to share my screen. Okay, welcome everyone to today's webinar. So my name is Audrey Parent. I'm an associate adjunct professor at UCSF. Our group there is interested in studying how breaks in immune tolerance leads to the development of autoimmune disease like type 1 diabetes. And we've actually been working with pluripotent stem cells as a model to look at some of these questions. So today I'm gonna tag team with Karla to talk about these topics highlighted here. So she's gonna start by trying to convince you why we need to have these stem cell-based platforms to model human type 1 diabetes, why they're so powerful to answer some of the questions that we're interested in. Then we're gonna go over some of the current state of differentiation of a few T1D relevant cell types. So she's gonna cover stem cell-derived beta cells and islets. And I'm going to talk about T cells and thymic epithelial cells. And then along the way, we're going to show you some examples of how we can use these stem cell derivatives to model disease. And then at the end, I'm going to talk a little bit about how we can start to combine different cell types, and in particular stem cell derived islets, with primary T cells to look at very specific aspects of T1D. And then at the end, we're going to do a discussion and Q&A. Okay. Okay, awesome.
Yep. All right, great. Thank you, Audrey. So I'm Karla Leavens. I'm an instructor, soon to be assistant professor at the Children's Hospital of Philadelphia. And I am involved in the Hearn group through Paul Gadou's lab, which I'm a part of, who's one in the CHIB consortium. And then myself, I'm involved in CBDS through the new investigator initiative. So as Audrey said, I'm going to start out by talking about autoimmune diabetes and why we need better human models enabled to better model it and advance our knowledge of it. So very simplistically, type 1 diabetes results from autoimmune destruction of the beta cells, which are in the islets of the pancreas, and they secrete insulin to maintain glucose homeostasis. And because of an autoimmune glitch, there's immune-mediated destruction of the beta cells, which results in beta cell loss, insulin deficiency, and ensuing hyperglycemia. However, years of research have really made clear that this is a much more complicated process. And much more prolonged than we initially had thought. So there is an initial hit causing beta cell damage, but whether this is due to beta cell autonomous defects or due to a glitch in the immune system, we don't fully understand. However, there is infiltration and damage of the beta cell due to T cells that are infiltrating. And then progression occurs from a combination of CD4 positive and CD8 positive reactive T cells, antibody producing B cells, as well as resident innate immune cells. This causes further destruction, insulinitis, beta cell death, and then ultimately dysfunction of the remaining beta cells that are there. And then you get, once you go below a certain threshold of beta cell mass, you end up getting a clinical onset of overt diabetes. Now, while we know that this occurs generally because of a combination of genetic risk and environmental factors, in each individual, why this exactly occurs may be different and from different cell types that are together. So this leads to the idea that type 1 diabetes is not a single disease, but may be the result of many endotypes or underlying disease processes that all result in the same consequence, which is beta cell loss, insulin deficiency, and ensuing hyperglycemia. Because there are so many cells involved, we really need good models to be able to model all of these interactions that result in this complicated disease. So we have some models that have been used to date. So we have beta cell and immune cell lines. These are easy to grow and genetically manipulatable, but they have variable function and it's hard to put them together to model this complicated disease. There are islets in primary immune tissues, and things like resources like Hearn have made these things more readily available for us to study, and advanced techniques have allowed them to be cultured for long periods of time so studies can be performed. However, even with increased accessibility, there's still a limited resource. There's unknown genetic and environmental influences from the individual donors, and there's a lot of variability that occurs. Moreover, islets available from type 1 diabetes donors are scarce because if the disease has been going on for decades, very few will remain, and those that remain may not truly reflect what is going on pathophysiologically and may be different from those that were lost during the progression of disease. Finally, we have many mouse models, including the non-obese diabetic mouse, and these are great to study whole body physiology, and they're manipulatable. There's actually an excellent review by many Hearn members recently in Molecular Metabolism as part of that review series that Joyce referenced about all the different humanized mouse strains that are trying to more closely resemble human immune system to be able to better model diabetes. However, even with these modifications, mouse models do not always recapitulate human physiology. An example of that is if you look at if you look at islet architecture, in a mouse cell on the bottom, all the beta cells, which are in red, are confined to sort of the core of the islet with all the other endocrine cell types, including alpha cells and delta cells on the periphery. Whereas in the human, you can see all of the different cells are mixed together. So the architecture and the physiology is intrinsically different, and we're not going to be able to fully achieve modeling that in mice. So all of these different models complement each other, but we really need further models to be able to further our knowledge about type 1 diabetes pathophysiology and strategies to develop further treatments. So a little bit of background on human pluripotent stem cells so we're all on the same page. So human pluripotent stem cells, or HPSCs, are defined by their ability to self-renew indefinitely and can proliferate without differentiating in culture for a long period of time, but then when directed can differentiate into any cell type of the 3 primary germ layers. Within the overarching category of HPSCs, there's embryonic stem cells, or HESCs, which are cells derived from the inner cell mass of a blastocyst. They were first generated from mice in the 80s and from human in the late 90s. And though there was a lot of scientific and political discourse about this, the NIH came up with definitive guidelines in 2009 defining the criteria under which cell lines that are used in NIH-funded research must have been generated. And all of the cell lines that can be used for NIH-funded research are included in the NIH Human Embryonic Stem Cell Registry, which currently includes about 500 different cell lines. The other cell type under HPSCs is IPSCs, or induced pluripotent stem cells. So these are adult somatic cells, either fibroblast or white blood cells, that are reprogrammed using 4 different transcription factors to a pluripotent state. And these were first described in 2007 by the Yamanaka and the Thompson labs. And while we are starting to describe the differences between IPSCs and HESCs, there still are differences that we don't fully understand. How the sort of workflow that we can then use these cells to disease model is, so again, we have our ESCs on the left in red derived from blastocysts. We have iPSCs that are reprogrammed from human blood cells. These cells can proliferate indefinitely in culture, but then when directed through differentiation, can generate ectodermal, mesodermal, and endodermal cell lineages. So when we think about using this for disease modeling, we can take a candidate gene that we identify in a patient, and we can make iPSC lines from that patient. Using CRISPR-Cas9 genomic editing, we can then edit that so that we have 2 isogenic lines in parallel that only differ with the difference of the gene that we are interested in. So we have patient and control isogenic lines. The complement to this on the embryonic stem cell line is taking an HES cell line, introducing that mutation through genome editing, and then we have a wild type and a mutant cell isogenic cell lines, and then we can use these for disease modeling by generating the different cell types that we're interested in putting together in a platform to be able to further investigate our disease. So getting specifically into the state of pancreatic beta cell differentiations, which is obviously very relevant for diabetes. So really the protocols that have been developed to generate pancreatic beta cells from stem cells or stem cell derived beta cells, SC beta cells, is really founded in decades of model organismal biology, specifically really mouse developmental biology. So this is an example from Doris Stoffers and Joel Habner's group, where they took LAC-Z under control of the PDX1 promoter. And you can see in a mouse embryo at day 9.5 that there is expression of PDX1 in the dorsal and the ventral pancreatic buds. And countless studies like this combined really help define the expression of the domains and gradients of the different transcription factors that define each region of the gut tube here. And gradual spatial as well as temporal mapping of all of these different pathways then allowed for the development of protocols so that we could then direct differentiation of stem cells into beta cells. So again, decades of research went into understanding that stem cells to be pushed to definitive endoderm require activation of TGF-beta nodal pathways as well as Wnt signaling. Subsequent differentiation from endoderm to pancreatic progenitors require inhibition of sonic the hedgehog as well as activation of pathways by retinoic acid, FGF7 and FGF10. And then further differentiation into endocrine progenitors and beta cells require inhibition of Notch, BMP, and TGF-beta signaling. So when we can represent this as sort of a linear progression from stem cells to beta cells, so this topic has been reviewed very extensively in a number of excellent reviews, including this one in Cell Metabolism that I reference. And you can see the different cell types labeled on the top as we progress through the developmental stages, the different transcription factors that are expressed in each of these different stages. And then on the bottom, the different pathways that have to be activated in black or inhibited in red in order to progress through these different stages. And these protocols really developed very consistently but incrementally over time until almost simultaneously 3 groups in 2014 and 2015 published very similar protocols almost simultaneously that still serve as the foundation of what we are able to do to differentiate stem cells into beta-like cells. And since that time, the goal has really become to make better, more mature, more functional insulin-like cells that more closely resemble their in vivo counterparts. So while initially only a couple labs could do this, now many, many groups across the country as well as across the world can successfully generate these cells for studying. So here in our lab, we use a couple different protocols. You can grow them adherently. You can grow them in aggregates. We grow them in small scale aggregates using low cluster plates on an orbital shaker that then over time generate these stem cell like beta cells. You can also scale up, and many groups use these shaker flasks here, such as Juan Alvarez, who is a HIRN member at the University of Pennsylvania. His group and others are currently trying to improve our protocols by studying, in his case, circadian rhythms that can further mature the cells and make them more functional as well. Additionally, we can do things such as reaggregating or sorting the cells, again, to sort of strive towards a more in vivo resembling cell. This is an example from Audrey's group where they took stem cell derived beta cells expressing GFP under the control of the insulin promoter, and then can specifically sort and reaggregate these to get more pure beta cell populations for study and manipulation. So using these protocols, we can take our panels of control stem cells, either ESCs or IPSCs, genetically modified or patient-derived IPSCs, push them all through directed differentiated to beta cells, and then study them by having them undergo various stresses, immune interactions, and then measure their properties or assess immunogenicity in order to further our knowledge of type 1 diabetes. So giving two specific examples, one looking at modeling beta cell autonomous defects. This is some data by Paul Gadou's group that's been published looking at HNF1 alpha, which is a transcription factor that causes monogenic diabetes, as well as been implicated in type 1 diabetes. As you can see, using HNF1 alpha heterozygote stem cells, we can make beta cells from these that are insulin expressing at a normal rate, but have higher degrees of glucagon expression as well, suggesting that there's a little bit of a developmental defect with HNF1 alpha deficiency. We can look at insulin secretion in response to low glucose and high glucose and see that with loss of HNF1 alpha, you get sort of dysfunction and blunting of glucose-stimulated insulin secretion. And then my current studies are looking at trying to understand how this could influence subsequent development of diabetes, such as doing things like decreasing mitochondrial number and how that may have an influence. So this is looking at very beta cell autonomous defects and how that could relate to development of diabetes and beta cell dysfunction. There's many groups through collaborations like Hearn that's giving access to type 1 diabetes donor and patient samples. This is an example from Doug Melton's group taking samples and making IPSCs from type 1 diabetic donors as well as normal donors, directed differentiating them into beta cells and alpha cells. And you can see that there's not a lot of difference in the yield of beta cells versus alpha cells in the normal donors versus the type 1 donors. And then you can take these cells and start performing more complex modeling, such as exposing them to stressors like thapsigargan and interferon gamma, seeing what HLA expression is. They go on to then show immune interactions between these and autologous PBMCs. So you can see that from each of these, we can sort of build and get into further questions of what are the interactions that are resulting overall in type 1 diabetes. So while these cells are really excellent and have made a lot of progress and really providing a complement model of what we've been able to do in the past with cell lines, primary tissue in mice, we also have to acknowledge that there are caveats and limitations that we have to work to overcome to really develop a better, more complete, more in vivo-like model. And as I mentioned before, we're working to make these cells more mature, making them more stable over time, as well as trying to resemble the composition of the pancreatic islet as well. There's also work trying to make these more heterogeneous to try to model more what we see in vivo and to understand whether this is important for type 1 diabetes modeling. And then, like many areas of biomedical research, there's some issues with consistency. So not just consistency between groups and between reagents, but these protocols need to be developed to be more robust so that we can get consistency across cell lines. So this is an example that I give, that I have done in collaboration with the Penn IPSC core, as well as the HPAP group within Hearn, where we take human embryonic stem cells, an iPSC line developed from normal donors, an iPSC line developed from a type 1 diabetes patient through the HPAP group. You can see through direct differentiation that these all make sort of similar amounts of pancreatic endocrine cells determined by their PDX1 and NKX6.1 double positivity. But when you continue to push them towards C-peptide and NKX6.1 double expressing stem cell derived beta cells, the different IPS lines have very different yields in them. And we need to improve our protocols to the point that we understand whether this is true biologic difference or whether this is just technical variability. So these are things that we hope to improve and strive to improve as a field to really make this a better and better model in which to investigate beta cell development, function, and the implications for pathophysiology. And then finally, the field has really been focused on the development of beta cells, but there's many different cell types, not just endocrine, but also exocrine and vascular and endothelial cells that reside in the pancreas. And we really need, in order to really develop platforms in which we accurately model type 1 diabetes and all the different cellular interactions, we need to be able to develop all of these different pancreatic cells, as well as all the immune cells that are involved in type 1 diabetes. And with that point, I will leave it to Audrey to give you guys an update on that compartment.
Okay, thank you, Karla. So let's switch gears a little bit, move away from the pancreas to now look at some of the other cell types that we think are relevant to include in our models for T1D. So since it's an autoimmune disease, one thing that we do need to consider is the immune system. And one way to do that would be to be able to generate hematopoietic stem cells from these pluripotent stem cells so that they would then reconstitute the whole immune system. So to give you an idea, this is the potential for HSCs to give rise to all of these cell types. I'm not going to talk about that too much today. This is not my expertise, but I do want to focus on one cell type that is very important, which is the T cell. So one thing also to notice from this diagram is that very early on, there's a branching point in the HSC potential between myeloid and lymphoid progenitor cells. And then lymphoid progenitor cells can give rise to both T cells, B cells, and NK cells. So we need to keep that in mind when we're thinking about how to actually make T cells from stem cells. So this is just a simplified view of how to get there. It's kind of the same idea as what we do with the pancreas. A lot of this is inspired from what has been learned from embryogenesis. And basically, we try to break it down into different stages that we reproduce into the dish. So in the case of T cells, they're not from definitive endoderm, they're from mesoderm. So the first step is different than when we make pancreas. Most of the protocols that are out there are 3D protocols. So they're going through what we call embryoid body formation. So doing the differentiation with spheres. Then the first step is to induce hematopoietic commitment. So this is going to be characterized by the expression of CD34 on these hematopoietic progenitors. And then if you remember the branching point, the next step will be to push them towards a lymphoid progenitor cell, which has been a bottleneck in the field for a really long time. So the HSCs derived from stem cells tend to be biased towards the myeloid branch. So people did a lot of work trying to understand how to get the lymphoid commitment to be more efficient. So what are the key signals at these two branching points? So one way of improving the lymphoid progenitor cell formation is to co-culture these hematopoietic progenitors with mouse stromal cells that express Notch ligand, so things like DLL1, DLL4. So the mechanism there is not clear, but it works. Some groups have done that very efficiently. And then there's also some work that has been done, mostly pioneered by the group of George Daly, showing that one important factor that's blocking the lymphoid commitment is EZH1. So this is a chromatin remodeler. If you knock it down at this stage, then you can improve differentiation into lymphoid cells. And then the second branching point where we really want to be able to control the differentiation is to push the cells toward T cells. And the key factor there, the key signaling pathways is Notch signaling, which we've known for a long time based on some of the work into mice and human T cell development. So in order to provide this Notch ligand signaling, there's different ways that you can do that. Historically, people have been doing 2D differentiation where you just culture mouse stromal cells that overexpress Notch ligand. And then you place the T cell progenitors, the lymphoid progenitors on top of these 2D culture and let them further mature. The problem with that is it tends to be biased towards non-conventional gamma delta T cells, which is not really what we want to make in these protocols. So in order to address that, some groups have developed a 3D differentiation approach instead, where they take the same mouse stromal cells and put them in a 3D organoid that they grow at the air-liquid interface. And that seems to promote formation of more conventional alpha-beta T cells. And then more recently, people have published stromal-free differentiation protocols where instead of using these cells, you can actually use plates that are coated with Notch ligand like DLL4. And that also seems to promote formation of conventional T cells. So this is a very broad overview. If you want more details, there's some really good papers out there. And the Hearn review that we've published recently actually cites a lot of these key studies. So I encourage you to look it up if you're interested in the actual differentiation process and what are the growth factors that are important. So I did want to show a few examples of what we can do with these T cells right now. Most of this work has been focused on cancer. And one thing to note is these protocols right now tend to give mostly CD8 T cells. So you can see in the fax plot here that most of the cells are CD8 and very few CD4s. So what people have been doing is mostly using these CD8 T cells for tumor killing assays. There's been some work with TCRs and some work with CAR. This is an example with TCRs where people have introduced an ESO TCR at the pluripotent stage. And then you differentiate the cells up to the T cell stage. And then what they've shown is, for example, you can take tumor cells that express the antigen that's recognized by the TCR, implant these cells into mice. And then if you inject your stem cell derived T cells, you can partially control the growth of tumors. So this is an example to show that these stem cell derived T cells are actually are functional, and they can target tumor cells based on the presence of the antigen. Another example that's a little bit more recent is in this case with a CAR. And instead of transducing at the pluripotent stage, what this group did is to transduce the differentiated cells. So they had them go through the differentiation process, use them to introduce the CAR. And then same idea, they took some tumor cells that express the antigen, in this case CD19, implanted them into mice, and then injected the T cells to see if they would be able to control tumor growth. And what you can see is that the black bar is the negative control. So if you don't have any T cells, these tumors grow really quickly, and within a short time frame, all of the mice die. Opposite, when you have the positive control, which in this case are PBMC derived, so primary T cells, it's the orange curve that's a little hard to see, but basically all of the mice survive, which means that the T cells can control tumor growth. And then for the stem cell derived T cells, they actually compared two different ways of making the T cells. And then the conclusion was that if you use this EZH1 knockdown procedure, which is the blue graph, then they work as well as primary T cells to control tumor growth, and all of the mice also survive. So this is exciting. Those are examples of application for these stem cell derived T cells. However, I want to point out some limitations of this process. The first one is the very low efficiency of CD4 T cell generation. So you can see in the bar graph here that it's very, very low. And also there's not really any regulatory T cells that are produced. So if you're interested in modeling something like type 1 diabetes, being able to produce Tregs is important. So that's a big limitation of the model. And then one thing also that I think is important to mention is that there's no proper education of T cells in this model. So it's not clear what the T cells are being selected on in these culture with mouse stromal cells, because there are no thymic epithelial cells in there. And that brings me to the thymus and why I think it's really important to pause and think about how can we actually make better T cells. So I think one key component that we need to include are thymic epithelial cells. And just to give you an idea of how T cells are normally formed, so they will go to the thymus. So the lymph node progenitors will migrate to the thymus. One of the first things that they actually see in the thymus is very high Notch signaling. So the Notch ligand, it holds true also for the thymus. And then they will go through this complex series of maturation steps where they will go from double negative to double positive CD4, CD8 expressing cells, and then single positive, either CD4 or CD8. And this process called positive selection normally involves interaction with a very specific subset of cells in the thymus called cortical tech or CTEC. Once you've managed to make this pool of T cells through positive selection, another important step that normally happens is what we call negative selection, when these T cells are going to be educated in order to learn the difference between self and non-self. And this is really important in the context of type 1 diabetes, because part of the issue is they forget that beta cells are self and they start destroying them. So the way that they're normally educated not to do that is in the thymus. And one example is by having a subset of techs called medullary tech or MTech present to them insulin peptides. And that way, if there's high affinity, for the T cells have high affinity for these insulin peptides, they should get deleted in the thymus. And then there's this also complex process that can happen if the affinity is lower, but not there's still some affinity, they can turn into Tregs, and then the cells that don't have any affinity for self-peptide can move on to become conventional T cells. So it's a complex process, but it's very important to keep in mind if you're interested in understanding what happens in autoimmune disease like type 1 diabetes. So in order to integrate this idea of thymic selection in our models, we think that it's really important to be able to make these thymic epithelial cells from stem cell. So our group, along with some other groups, have spent quite a bit of time developing these in vitro differentiation protocols. So this is the overview here, simplified view of how it works. So the thymus is also derived from definitive endoderm. So the way we started with this is actually by taking what had been developed for the pancreas and adapt it to make a thymus instead. So instead of doing the pancreatic bud, then you would push the cells to be more towards the anterior foregut and then pharyngeal endoderm, and finally push them to become thymic epithelial progenitors. And then one thing to note is that the last step of maturation, where you go from progenitor to fully functional thymic epithelial cells, requires signals from T cell progenitors. So it's actually pretty hard to do in vitro. If you want to know more about how to do this differentiation, there's actually a good review that was published last year, which I suggest you look it up if you want to know more about the differentiation factors. Okay, so I wanted to show a little bit of data just to give you a sense of where we stand in terms of the efficiency of differentiation for this cell type. So this is data from our group. So the way that we do it is we do the first stages in suspension. So we do 3D differentiation originally, and then at some point we plate the cells in 2D, and that triggers further maturation into thymic epithelial progenitors. Then we can look at different markers. This is an example looking at cytokeratin 5 and 8, which are typically expressed in MTEC and CTEC. And what you can see is that in our differentiation, it's still a little bit heterogeneous, meaning that there's some islands of cells that have the right markers, but there's also some other cell types. So it's not as efficient as with the beta cell differentiation, which most of the culture usually go into the pancreas lineage. We've also acquired some new tools. So we now have a reporter line. So similar to the insulin GFP that we use for the pancreas, we have a FOXN1 GFP for the thymus. So all of the cells that turn on this really critical transcription factor, FOXN1, will also turn green. So we can actually quantify exactly how many cells are going into the right lineage in our culture. So this is for in vitro. Then to assess the function of these cells, to see if they can actually support the development of T cells, we've also been working on different mouse models. Just want to talk quickly about this one, which is actually supported by HIRN. So it's part of our grant in CMAI consortia. So in this case, we work very closely with the group of Megan Sykes at Columbia University, where basically the cells are differentiated in vitro, and then they get shipped to Columbia, and they do all the in vivo work. So what they do is they take NSG mice and they thymectomize them. So they get rid of the endogenous thymus to be sure that if we see some T cells appearing in these mice, we want to be sure that they're actually coming from our stem cell graft, not from the endogenous thymus. And then they irradiate these mice and inject human CD34. So for now, these HSCs we use primary HSCs, but at some point, the goal is to use stem cell derived HSCs instead, so that everything is going to be matched in the mouse. And then when we send them the cells, we've done different ways of doing the transplantation, but so far what seems to work the most efficiently is to use a living scaffold. So we actually use a swine thymus scaffold that we inject our stem cell derived thymic epithelial cells in there. And then once you transplant them, you can harvest the tissue and find your cells again by doing some specific staining. So for example, we use human class 2, HLA class 2, and co-stain with cytokeratin, so that when we see these yellow cells in the tissue, we know that we're looking at human techs. Okay, and then there's some assays that we've also developed there to really be able to measure the impact of these human stem cell derived thymic epithelial cells on positive and negative selection of human T cells. So Megan's group has all these tools. Basically, they have specific TCRs that are not getting positively selected in swine thymus. And then when you inject functional human techs in there, then you start seeing these TCRs go through the selection process. So we can actually read out function of our human cells. And then how do we actually use this to model T1D? Well, the next step will be to actually do patient IPSCs to see if there's some defect in their ability to support human T cell development. And also we can specifically modify some genes that are involved in thymic selection. So for example, we're interested in the role of air, and we can do that in our HESCs instead of IPSCs. Okay, so just a quick word about the limitations of these the stem cell-derived thymic epithelial cell process. One thing that I wanted to say is that the maturation of these cells, as well as the maintenance, is pretty complicated and takes a long time. So if you're thinking about doing these experiments, we actually have to leave the cells for 20 to 25 weeks into mice. So that takes a really long time. So it's a clear limitation of the system. And then also the heterogeneity and complexity of the tissue. So when I started working on this, we thought we would have to make two cell types, CTEX and MTEX. Now with all of these new technologies, doing single cell RNA-seq and really digging deep into what is found in a human thymus, we realize that it's a lot more complicated. So MTEX are actually made of a lot of different subsets. And there's this nice review that was recently published, I think, that highlights the complexity of the thymus. So we'll see. I think the jury's still out to see if the stem cell-derived thymic epithelial cells will be able to reproduce all of this complexity, but it's an interesting system to actually look at that. Okay, so for the last part of the talk, I want to highlight some of the examples that we can actually do with the tools that we have. So we've gone through the current state of the differentiation and what are the limitations, but there are some things that we can actually do today. And one of the steps I think that we can focus on is looking at islet infiltration and beta cell killing by T cells. And I want to show a few examples of that. So why have we not been able to do that before? What are the actual bottlenecks that prevent us from doing that? Well, one of the problems is the requirement to match HLAs between beta cells and immune cells. So if we want to be modeling destruction and not alloimmune destruction, we need to make sure that there's a match between the HLAs of beta cells and immune cells. And one elegant way that people have been doing that is by having donors give cells to make IPSCs, and then having the same donor give blood so that you can actually do isogenic co-cultures between iPSC derivatives and the T cells. One way that I think we can also do that, which is a little bit easier technically, because you don't, when you do the isogenic system, you have to use PBMCs from that one donor. But if instead, what we can do is selectively delete some of the HLAs from our favorite cell line. So for example, we've done this with our INSGFP cell line, where we deleted all of the HLAs but HLA-A2. Then when you make beta cells from this cell line, they only express HLA-A2. So that makes it a lot easier to match with PBMC donors. So you only have to worry about this one HLA. So I think that's an alternative to this isogenic system. And then another bottleneck that I think is making the system a little bit less robust is the low frequency of autoreactive T cells. So there's not a lot in the blood, even if you look at PBMCs from patients with type 1 diabetes. So one way around that is to actually isolate T cells from these donors and transduce them with your TCR of interest. And then you can actually look at a much more frequent population of autoreactive T cells. Once you've figured out the cell source, then there's different ways that you can co-culture them, that you can put them together. And there's actually a lot of really interesting work that's being done through Hearn on all of these models. For example, in our group, we're interested in doing in vitro co-culture. There's people who do microphysiological systems where you can now introduce endothelial cells and have a little bit more complex system. And then also in vivo humanized mice. So just to give you a quick example with the in vitro co-culture. So this is data from our lab. So we take our stem cell derived islets, and then you can co-culture them on their own, which is what is shown here. The cells are green, so that you're looking at insulin expressing cells, and then they're pretty happy on their own. And then when you add some T cells, so you can add, for example, wild type T cells, which have not been manipulated. They're just isolated directly from the blood. And when you co-culture them, the stem cell derived islets are not being destroyed. So they're pretty happy. There's a ton of green there. However, when we transduce these T cells with specific TCRs, then you can see the cells, the green going away. So the beta cells are being killed by the T cells. So it's a pretty powerful assay. Now that we have this set up, we can start looking at specific questions related to type 1 diabetes. So for example, we can do genetic modification in the stem cells, in the stem cell derived beta cells, and try to see how that affects the immune response. You can do the same thing in T cells. You can look at different TCRs. You can also think of adding, activating, and blocking molecules in the co-culture to see how manipulating these pathways will affect the immune destruction. Okay, so this is in vitro. If you want to go a little bit more complex, so you can go towards microphysiological systems. So this is one example from a group from Hearn. So what they've done is they put together islets, endothelial cells, and stromal cells in these chip. And then you let the endothelial cells and the whole system organize. And then what you end up with are these nice blood vessels in the chip and the islets, which are embedded within it. And then you can do a lot of different assays. You can look at beta cell function over time, but you can also start adding immune cells in there. And for example, they've added CD3 positive T cells, and you can really see these cells move through the blood vessels and then infiltrate the islets. So that allows you to actually look at a little bit more complex questions like migration and which factors affect that. And then just quickly wrap up, I want to talk a little bit about humanized mouse models. There's also quite a bit of work done through Hearn on these models. This is one example where we use PBMCs. So basically, you can use NSG mice, or you can use some of the newer substrain that have been developed. So this is a really nice paper by Mike Bram's group, where they basically knocked out murine MHC class 1 and class 2. And what that does is when you inject human T cells in these mice, they are not going to attack the mouse tissue and cause what we call GVHD. So the mice can actually, you can keep them much longer and have a much bigger window to do your assay. So what you can do in terms of modeling is inject either directly PBMCs, or you can isolate T cells and introduce whatever TCR you're interested in and put these T cells back into the mice and see if now you can actually see autoimmune destruction of beta cells. So that's one example. And then another example, which is a little bit more complex to set up, but this is something that also is being developed through Hearn, in part by the group of Megan Sykes. So in this case, they use regular NSGs that are thymectomized, and then you inject them with human HSCs, and you provide a human thymus in order to reconstitute the immune system. So the thymus will provide robust T cell reconstitution, and then the HSCs will also allow you to have some of the other immune cell types. And then if you want to turn this into a T1D model, you can take some of these T cells, introduce a T1D relevant TCR, and inject the cells back into the mice. So now you have a higher frequency of these autoreactive T cells. And then if you want to look at destructions of human beta cells in this model, one thing that you can do is use HLA-edited stem cell-derived islets. So for example, if we use our HLA-A2 only cells and combine with HSCs and thymus that are HLA-A2 positive, then you're not going to get allo rejection of the islets, and you can start looking at autoimmune destruction of the beta cells in these mice. Okay, so if you have one thing to remember from the webinar, I think is that it's an exciting time to be working on these kind of models. I think if you combine advancements in stem cell differentiation with all the tools that we have in terms of genome engineering, this has really unlocked new opportunities to model T1D. And then one quick word about the Hearn review series. So this is what has been published so far. And I wanted to highlight the one that we've published, which has been the inspiration for this webinar. Had a lot of fun running this with Karla, Juan, Linda, and Holger. So if you want more information on what we've talked about today, I invite you to go read that review. And then final slide, just want to acknowledge our groups and people who have contributed to some of the data that we've shown today, and funding sources. Thank you.
Excellent. Thank you very much, Dr. Parent and Dr. Leavens, for a terrific talk. And now we'll take questions and answers and discussion. So if anybody has anything they would like to ask, do you want to raise your hand and come off of mute? Do you have any questions? Yes, I see from France Boot.
Yes, hello. Hi, Fransje here from the Netherlands. I'm a PhD student at the LUMC. Very interesting talks. Thank you so much. I have a quick question mainly about the last model you just discussed. I know that quite a few of the protocols we have currently that steer towards beta cell differentiation also give us alpha cells. Do you think it would be it would have some worth to also include the alpha cells in the further onset type 1 models? Because we know that alpha cells will survive an initial immune attack, and perhaps it could be interesting to study the dysfunction of the alpha cells. And I feel like right now we're just sort of putting them to the side. So I was wondering what your opinion on this is.
Yeah, I think that is an excellent point. You know, I mean, the protocols to this point have been very beta cell centric because it is the clear cell involved with type 1 diabetes, but there are actually specific protocols in the last 5 or 10 years to specifically differentiate alpha cells as well. So I think people, like you suggested, are increasingly recognizing that beta cell, the type 1 diabetes is not just a beta cell and an immune cell disease, that we have to include the other endocrine cells within the pancreas. So those protocols for making alpha cells, there's a couple coming up for making delta cells to be able to differentiate them and then have a much more sort of in vivo model of an islet is definitely, I think, the direction that the field is moving into. But we're definitely playing catch up with the other cell types.
Yeah, I agree. And one thing I want to add is it's a little challenging with alpha cells because you're always going to have the endogenous alpha cells in these mice. People have been doing STZ to kill the beta cells, but I think there's now glucagon knockout mice that exist that you can actually use to transplant your stem cell-derived islets. So you can actually more look into alpha cell function more specifically.
Yeah, that's a great one. I haven't seen people transplanting too much of sort of all of islets back into mice. It's really sort of ex vivo at this point, but certainly that's direction I think that everyone hopes to move.
Great, thank you.
Thank you.
Christina?
Hi, good morning, everyone. Thank you so much for this. It was very interesting. I have a question about the thymic epithelium and the stem cell derived one. I'm not super familiar and up to date. I was wondering, Audrey, you mentioned it takes 25 weeks to see the development of these cells. This is after transplanting progenitors. And my question is like, you're questioning whether you're giving rise to all the different populations. Have people looked at this, and can you generate all the different cell types of the thymus, or are they biased to a particular cell types?
Yeah, so far our analysis has been mostly focused on just the basic C-Tech versus M-Tech. I think all of these different subset is so new. So we've just in the last, 3-4 years, maybe now realized how many more different subset there is. We've been working on building panels, immunofluorescence panels, to look at some of these subsets in the human thymus. So I think it will be really interesting to see now that we have a lot more tools to look at them when we pull out the tissue from the mice, refine our analysis to see if we only make like one subset of M-Techs or if we make multiple of them.
Thanks.
Thank you. Iji Yoshihara, you have a question?
Hello. Can you hear me?
Hello?
Yeah, it's a very nice talk and it was very impressive. So I would like to ask you your opinion. As you know, the T1D is quite kind of slow progressive diseases. And then current stem cell derived islet and co-culture system in the T1D PBMC, actually they show the quite quick response for the beta cell killing compared to what we see in the in vivo in human. Do you have any idea to recapture this actual slow progressive T1D using your system, or any other idea? Do you have any?
Yeah, so I'm not sure I understand fully. So you're saying that if you use T1D PBMCs to reject stem cell derived beta cells, that it's faster than
Yeah. So in vitro, actually, we are actually doing the similar stuff. We actually see the beta cell killing in in a few days. Actually, that's happening when we co-culture with the T1D PBMC with the stem cell derived beta cells. But in human, in vivo, in my understanding, the progression of the T1D is quite slow, right? It's not gonna happening in one day. So do you have any idea why we see this discrepancy in vitro and in vivo?
Yeah, I guess the immune system has a lot of different layers, which are hard to recapitulate in vitro. So in vivo, you will have a mix of innate immune cells, CD8 T cells, CD4 T cells. You have some Tregs that can also kind of put a brake on the whole thing. So I think in vitro, we have to pick like which part of the immune response we want to look at. So we might not be getting like the full picture.
Yeah. Okay. So that's the one question. And then do you have another question? Do you have any idea for what's the best model currently so far available in vivo model? For example, a humanized model mice. As you talked about the PBMC, NSG PBMC is one of the model, but they don't survive that much long, right? We can actually do only 1 month, 2 months experiments.
But I think with the new substrains, so we've now switched to these NSG mice that are missing murine class 1, class 2.
Mm.
So they actually survive much longer. So that has helped with lengthening the experiments.
I see. So they live like 3 months, 6 months, or that much long.
Yeah, so we've been able to keep them for a while.
Okay.
And then this paper that I referenced, they actually also characterize really well the what happens when you put T cells and just wait.
Okay, thank you.
Great, thank you. I have time for one last question, if there's any. Okay, I don't see any other hands raised. So we want to thank Dr. Parent and Dr. Leavens again for a very wonderful talk. And remember, it will be on YouTube on the Hearn website very soon. So thank you all for coming today and hope to see you again at a future webinar. Thank you. Bye-bye.
Thank you.